Some fungal infections can become life-threatening, especially when the immune system is weakened, and the problem is getting worse. Many fungi are becoming resistant to existing medicines, while relatively few new antifungal drug classes are being developed. One of the strongest weapons doctors have is a drug called Amphotericin B. It belongs to a family of medicines called polyene antifungals. These drugs can attack a wide range of fungi and have remained useful even after decades of clinical use. But there is a serious problem. Polyenes can also be highly toxic to human cells, so scientists face a difficult challenge. How do you preserve the fungus-killing power of these drugs while reducing the damage they can cause to the patient? Now, researchers may have found a new answer. Instead of building an entirely new antifungal drug, they used enzymes to redesign existing ones, adding sugars, changing chemical groups, and creating compounds that were more powerful, less toxic, and easier to produce. One of these redesigned molecules reduced a dangerous fungal infection in mouse lungs by 87%, but the most surprising discovery was still to come. The new compound may no longer kill fungi in the same way as the original drug. To understand the discovery, we first need to understand why polyenes are both effective and dangerous. Fungal cells are surrounded by a membrane containing a molecule called ergosterol. Human cell membranes contain a related molecule called cholesterol. Polyenes, such as Amphotericin B, interact strongly with fungal membrane sterols. This disrupts the fungal cell's protective barrier and contributes to its death. Unfortunately, the drug can also interact with components of human cell membranes. That helps explain why Amphotericin B can be effective against severe infections, but still produce serious toxicity. Scientists have tried to redesign these molecules before, but polyenes are large and chemically complicated. Changing one specific part of the molecule often requires many synthetic steps, protective chemicals, and expensive reactions. Some promising Amphotericin B derivatives have taken more than 10 chemical steps to produce, with extremely low overall yields. The researchers behind this new study tried a different approach. They asked, could biological enzymes modify these drugs more cleanly and efficiently? Enzymes are natural molecular machines. Inside living organisms, they build, cut, and modify molecules with remarkable precision. The researchers searched bacterial genomes for enzymes that could alter polyene antifungal drugs. They discovered several unusual enzymes called glycosyltransferases. These enzymes attach sugar molecules to other compounds. Most polyene antifungals already contain one sugar that is important for their activity. But the researchers found enzymes capable of adding a second sugar. One enzyme, called KFUSV, was particularly flexible. It could attach different sugars to several polyene molecules, including Amphotericin B and nystatin. Adding the second sugar improved certain properties of the drugs, including antifungal activity and solubility. But there was a problem. The additional sugar alone could also increase damage to red blood cells. So the researchers introduced a second modification. They used another enzyme to change a carboxylic acid group that contributes to the toxicity of many polyenes. This created a series of dual-modified antifungal compounds. One change added a second sugar. The other replaced or altered a chemical group associated with toxicity. The combination produced some of the study's most promising results. The scientists tested the redesigned molecules against 10 clinically important fungal pathogens. These included fungi considered critical or high risk, as well as strains resistant to existing drugs. Some of the modified compounds were more effective than their original parent molecules. One derivative, called NIS-33, was reported to be more potent than Amphotericin B against an azole-resistant strain of Aspergillus fumigatus. Other modified molecules showed improved activity against certain Fusarium species and an Amphotericin B resistant strain of Candida albicans, but greater antifungal power would be useless if the compounds became even more toxic to human cells. So the researchers examined whether the second chemical modification could restore safety. The results were encouraging. Several dual-modified nystatin derivatives damaged fewer red blood cells than Amphotericin B and the unmodified compounds. One molecule stood out. Its name was NYS34. NYS34 contained both major structural changes, an additional sugar, and a modified carboxylate group. When tested against 3 human cell lines, NYS34 showed between 3-fold and 8-fold lower toxicity than Amphotericin B. The researchers tested cells representing the kidney, lungs, and liver. That does not prove the compound will be safe in humans, but it provided an encouraging early signal. NYS-34 was then given to mice to study tolerability and how the compound moved through the body. The researchers found that it reached several major organs, including the lungs. At higher repeated doses, the mice eventually showed signs of toxicity. That was important because it helped the researchers estimate a safer experimental dose. They then tested NYS-34 in mice with an invasive lung infection caused by Aspergillus fumigatus. The mice had weakened immune systems, making the infection more similar to the dangerous fungal disease seen in vulnerable patients. The results were striking. NYS-34 reduced the fungal burden in the lungs by 87%. The mice receiving that regimen showed no obvious signs of toxicity during the infection experiment. In the comparison group, Amphotericin B reduced fungal burden by 35%, although that reduction was not statistically significant under the conditions of this study. This does not prove NYS-34 is superior to Amphotericin B. The doses, schedules, and experimental conditions were different, but it does show that the redesigned compound was active inside a living animal, not only in a laboratory dish. Then, the researchers examined how NYS-34 was killing the fungi. Traditional polyenes, such as Amphotericin B and nystatin, are strongly associated with ergosterol, the sterol found in fungal cell membranes. But when scientists tested NYS-34, they found no evidence that it bound ergosterol in the same way. Even when NYS-34 was pre-mixed with ergosterol, its antifungal activity did not change as expected. This suggests that the two structural modifications may have done more than reduce toxicity. They may have fundamentally changed the drug's mechanism of action. NYS-34 still killed fungal cells, but it may have been doing so through a different biological pathway. Researchers do not yet know exactly what that pathway is, and that unanswered question could be extremely important. A different mechanism might help scientists develop antifungal drugs that avoid some of the toxicity and resistance problems associated with existing treatments. Although the results are promising, NYS-34 is not an approved medicine. The majority of the experiments were performed using fungal cultures, red blood cells, and selected human cell lines. The infection study involved mice, not patients. Researchers still need to understand how NYS-34 kills fungal cells, whether it remains effective during longer infections, how the body processes it over extended periods, which organs may be affected, and whether it can be administered safely to humans. The compound also caused toxicity when mice received a higher repeated dose. That means the difference between an effective dose and a harmful dose still needs careful investigation. Before any human trial, researchers would need extensive testing of dosage, delivery, metabolism, and long-term safety. This discovery is important for more than one experimental drug. It demonstrates a new way to improve complex natural medicines. Instead of relying entirely on long and expensive chemical synthesis, scientists can use enzymes as precision tools. They can add sugars, replace harmful chemical groups, change solubility, reduce toxicity, and possibly create completely new mechanisms of action. As drug-resistant fungal infections continue to rise, that flexibility could become extremely valuable. So, have scientists created a safer replacement for Amphotericin B? Not yet, but they have shown that one of medicine's oldest antifungal drug families can be rebuilt, and by changing only 2 parts of the molecule, they produced a compound that was less toxic in early tests, active against dangerous fungi, and effective in an infected mouse model. Subscribe to BioFacts for more discoveries changing what we know about medicine, disease, and the hidden biology inside the human body.